I'm Helen White Cooper, and I would love you to apply to my project, which is all about localization of RNAs within cells. And I expect you know something about how important RNAs are in terms of getting information out of the nucleus into the cytoplasm, making proteins. But most cells are polarized, and so the proteins are made not just anywhere in the cytoplasm, but in one particular place. One extremely polarized cell that I bet you don't know much about is the sperm. And fly sperm are extraordinarily long, and at the point at which they express the RNAs that you would work on if you came into my project, the sperm of the fly is about 1 millimeter long, and I'm going to scale that up to the equivalent of a human, and here is the full length of that cell if you scale it up to the equivalent of a human. And the RNAs we're going to work on are made in the nucleus, which is at this end of the cell, and they're made there, and they move all the way along to the other end, to the very tail of the spermatid as it's developing, and then they're used to make proteins just here. And there's your newly synthesized protein. It's about 1 millimeter away, or on the scale of this shoelace, 1 meter away. What I want to know is, how do these RNAs get from here to here? How do they make the proteins here? They don't make the proteins while they're on transit, they make the proteins when they get there. There are particular RNA binding proteins that are almost certainly the anchors that hold them in place here. How do they recognize those RNAs? How does the protein make a gradient? Some proteins make a gradient that's really short. I don't have a longer prop, but some of them make a longer gradient. Some of them make a protein distribution that's really smooth. Some of them make it really speckly. If you were to come to my project, to work on my project, you would do lots of microscopy. You would take lots of pictures. You would really characterize what this protein localization looks like, what the RNA localization looks like, and how that whole process is regulated. You'd learn microscopy, you'd learn some mathematical modeling. We have a collaborator in physics who will do the maths with you, and you'd learn some really excellent biochemistry to understand how the RNAs and the RNA binding proteins work together to control this truly extraordinary bit of biology. Please apply. Thanks.